Thanks for inviting me and letting me speak again. I'm going to start with this slide, which is actually a slide I showed last year. Basically just to sort of emphasize again that obviously the brain is an important organ to be looking at in CFS-ME, and I think you has reminded us of that fact yesterday a couple of times. And one aspect in the brain that you can look at is the morphology, or the size and shape of the brain. And that has been done over the years, not quite as much as you would expect, but there are a couple of findings. The earliest ones I could find with this particular methodology going back about 12 years now from Okada. The problem with these findings are that they are quite diverse. It's actually very hard to find results that have been replicated across different studies, and there's probably various reasons for that. But we see sort of various findings with reduced gray matter in certain areas, no changes in white matter, then global reductions in gray matter, no changes locally, and so on and so on and so on. Then other findings that show no changes in gray matter, but changes in white matter, and so on. So it's a rather sort of diverse field, and one of the latest sort of findings just came out, was just accepted, I'm not sure it's even out yet, from Marika van der Schaaf, who presented here last year on a related aspect. She has probably one of the largest samples so far in this group, with almost 90 patients in that study, and also found no regional gray matter differences. She found some associations with pain again in the prefrontal cortex, but in terms of volume change as a difference between groups, there was no difference. So the aim of this analysis, of this study, was to add to the confusion and investigate differences in gray and white matter between volume between ME/CFS patients and healthy controls using modern Voxel-based morphometry methods. And since I'm aware that probably only a very small minority of you will be familiar with these methods are, I thought I'll spend a minute or so to talk about how these methods work. So we start out with our anatomical MRI scans that look something like this. And the first step is this step called segmentation, where we basically segment the images into areas that we believe are gray matter and areas that we believe are white matter. And we can do this for cerebrospinal fluid and other tissue types as well. And obviously, if we want to say we do that, it's a software that does that for us. Once we've done that, we then warp the individual segments towards a common template, so that basically brains from like two different or multiple different individual people and their segments now, after the warping, look far more similar to each other, and that's basically is designed to aid the statistical analysis on a voxel basis. So that step then is to simply smooth this out a little bit. This is primarily thought to sort of reduce the sort of uncertainty in the normalization. So the normalization isn't perfect, so we need to basically sort of, smooth this out a little bit and aid in the statistics. So what then happens is, so what we end up at this point here is with a map for each individual that roughly tells us how much volume of this particular tissue type is in that particular location, and those locations roughly match between the different individuals, despite the initially quite different morphology, of course. And then we can do group comparisons, and we can look at relationships with other variables. In this analysis, we included 42 patients, about 3/4 of them women, mean age 45 years. They all fulfilled the criteria. We, as Julia said yesterday, excluded any comorbid psychiatric conditions. So these were all participants in the MRC funded study from Julia Newton. here in Newcastle. Controls were 28 healthy controls, roughly the same sex and age distribution. 10 of those came from that same study. The remainder, so 2/3 of those, came actually from a different study that I was involved in, ran at the same time, but they were done on exactly the same scanner with very much the same sort of general distribution in terms of age and sex, so there's no difference statistically between those. Scanning to take in a 3D scanner in our magnetic resonance center, all very details of the pre-processing of this segmentation normalization was done with this computational anatomy toolbox. It's sort of the latest generation of a fairly respected tool in the field. Formerly, that was called the UBM toolbox. And we did the voxel-based statistics with including total intracranial volume, age, and sex as covariates to account for any effects that those might cause. What are the results we found? So in terms of overall brain volumes, so that's one thing that you sort of automatically get out of the analysis, is we saw a significant difference in total intracranial volume between the patients with lower volumes in patients and also lower white matter volumes in the patient group. But since the sort of overall total intracranial volume is really just a measure of head size in a way, so it's not particularly interesting. So we happened to recruit a patient sample that had slightly smaller heads. What we needed to do is simply correct for head size by dividing all the segment volumes by head size, and when we do that, we actually see a significant difference in gray matter volume, with actually increased gray matter volume in the patient group, and the decrease in the white matter volume remains significant, no difference in cerebrospinal fluid volume. Regionally, we see differences in gray matter increases in the patient group in a couple of reasons that are quite interesting in relation to the previous talk, because they're, I think, some of the regions that Julius sort of at least implied, if not mentioned directly, and they are in 4 clusters in total here, shown here. Biggest cluster is a cluster in the posterior insula on the right-hand side, extending into the right amygdala. Another cluster is mostly centered on the left amygdala, extending somewhere into the central structures, which isn't quite clear what those are. And then a third cluster in the medial temporal lobe. Middle temporal lobe. On the white matter side, as I said, we saw the reduction in the patient group, and that seems to be centered primarily on structures in the midbrain and brainstem here, and with a third cluster in the anterior temporal lobe right here. There's a second view of that just in these sagittal slices. So again, it's a midbrain and brainstem, specifically the pons. So what does that all mean? Yeah, in terms of the gray matter differences, I think those are areas that we would typically associate with things that are related to sort of interoception, so perception of internal signals, so specifically the insula, and also with the processing of salience and balance, so sort of like judging stimuli and whether they are important and whether they are good or bad for you. And that's sort of the role of the amygdala. The thing about them is that none of the previous studies have reported any of these areas before. So I think we need to be a little bit careful about not over putting too much emphasis on them until we've sort of replicated some of these findings. In the white matter differences, like I said, we said reduced volume in cortical spinal tracts and the brainstem pons that we heard from Julius talks of the role of the brainstem in sort of pain perception. So that's certainly seems to be an important region. And perhaps importantly here is that those are also regions that have been shown to be associated with CFS previously. So this is a study from Leighton Barton's group in Australia, and they had shown, for instance, an association between white matter volume and fatigue duration in very, very similar regions of the brainstem and midbrain in their study, and also a differential association between patients and controls between the signal, this is not volume, the T1 rated signal in the brainstem and the pulse pressure. Similarly, last year we saw a talk by Jose Montoya, and their group published this paper also last year, and they talked about various different findings. One of the findings they mentioned is that there's a reduction in what's called fractional anisotropy in one part of the fibers that they investigated, and they speculated that those perhaps could be related to a reduction of crossing fibers in a particular region. So that's one of the findings. So you see this fiber tract here is the inferior longitudinal fasciculus, and here in this region they saw this increase in fractional anisotropy. Interesting enough, that's exactly the region where we see this reduced volume of the sort of the uncinate fasciculus, which presumably is where we see the reduction in volume, where these two fiber tracts sort of come together. So that, again, that might point towards the fact that we might have sort of indirect evidence, at least, that reduction in crossing fibers may indeed be happening in this case. So, in summary, I think we've seen some gray matter differences that are interesting, but I think need to be replicated. Some of the white matter differences that we see in this particular sample seem to be related to some of the earlier findings, so they're certainly worth exploring further. But there are, of course, a number of limitations that need to be mentioned. I've already said the replication is going to be important to see if especially for the gray matter differences, if those are shown in a different sample, perhaps in a larger sample. And what's really important here is, I think, methodology needs to be very consistent. So even though voxel-based morphometry has been around for some time now, it's still a relatively young field and a young method. So, and there's quite a number of sort of developments in terms of the software that is doing it, and there's also sort of a number of changes you can make or choices you have to make as an investigator for, like, in the analysis pipeline, that I think we would want to be sort of consistent and make sure we're doing the best choice there, and consistent at least. We've heard this a long time, or often already throughout this conference. This is a cross-sectional study, so we don't know if these changes, assuming they are real, or differences are the result of the illness itself. So are they the cause? How are they related to the symptoms? It's very difficult to say from just a cross-sectional design like this. And the last thing might be a bit more obscure and not quite as obvious. One of the things that we always have to keep in mind with MRI images is that they are based on, or the segmentation in this particular case is based on the image contrast, and the image contrast really tells us about, like, the magnetic properties of the tissue. And there are other processes other than volume change that may influence these magnetic property tissues of the tissue, and they may sort of be perceived or may show up as a volume change, but may not in fact actually be a volume change. So we need to be careful and sort of investigate these further and make sure it's a volume change and not something else. And with that, look, I want to thank my collaborators on this project, the CMRC, obviously, for funding Newcastle Upper-Tyne Hospitals Trust for funding me at the moment, partly funding, and of course the study participants who volunteered their time for this.
Great. Thank you very much. Thank you for keeping to time. I'm afraid we have no time for questions. Sorry, you'll have to get on a coffee. Right. Now we've got another Andreas. It's very confusing. Andreas and Andrea from Brussels. Talking beautiful slides I've seen, immune pain interaction.
Bottom one,
That's not my presentation.
yeah, bottom one, and then it'll go down to yours.
So this
There you
one
go,
has to
that's
go
it.
forward.
Yeah, great. Okay, thanks for inviting me to present the poster. And I had prepared a couple of jokes about my accent, but then they told me I only have 10 minutes, so straight to the point. And so what I'm talking about today is immune pain interaction following exercise in chronic fatigue syndrome. We think this topic is rather interesting as we are the first that try to explore this in chronic fatigue syndrome, but also in other chronic pain conditions. And briefly why we think that pain is important and worth studying in chronic fatigue syndrome patients as well. It's first of all the second most important symptom for people with CFS, according to people with CFS, and sometimes it can also be even more distressing than fatigue. And actually CFS overlaps of about 7% with fibromyalgia that, as you know, is a condition characterized by widespread disabling pain. And a nice recent review on the topic showed that pain and fatigue share many underlying mechanisms, so maybe understanding pain better can also shed some light on chronic fatigue syndrome and fatigue mechanisms as well. Why we chose complement system and elastase activity as a measure of immune system activation is because systematic review on the topic and our own data as well show that interleukins that have been thought to be big players in the immune system activation are probably not able to explain CFS symptoms nor exercise-induced symptoms changes. And on the contrary, complement system and elastase activation can be part of these immune system alteration or activation, and they have been shown to be associated with fatigue and pain, and also to exercise-induced pain. Unlikely in animals, a recent study from 2009 showed that C4A more than C3A and C5A protein is related to pain in chronic fatigue syndrome, and it actually increased 6 hours after submaximal exercise test in humans. So what we did is that we enrolled 22 people with chronic fatigue syndrome and 22 healthy sedentary control. And if they were willing to participate, we asked them to come to the hospital twice. And the first time we asked them to perform a submaximal exercise test. That is a standardized test known also as aerobic power index. So they have to cycle at a fixed pedaling rate, which is around 60 to 70 cycles per minute. And while we increase the resistance that the bicycle gives to the subjects minutes by minutes, 25 watts per minute. And of course, and they have to stop, the exercise stops when subjects reaches 75% of target heart rate. Of course, before and after the exercise test, we measured some pain pressure thresholds that, as you know already, thanks to the previous presentation, we think is a measure of central sensitization or central nervous system hyperexcitability. And we also took, before and after the exercise, we took blood samples so we could measure complement system and elastase activation. And we did, after 1 week, okay, we asked them to come back again, and we did exactly the same procedure. So they underwent another exercise test, but this time we tried to make an exercise that would have not worsened patient symptoms. So the exercise was self-paced and physiologically limited. That means that we asked patients, how long do you think you can cycle today? And we also measured heart rate, and we also measured the anaerobic threshold so that we could stop the exercise as soon as we think it could have worsened their symptoms. And the results, we have a number of results, and I think one of the most important is that PPT responses are different between healthy controls and chronic fatigue syndromes. You already know that. And we confirm that in healthy controls, the pain thresholds actually increase after an exercise. So we need more pressure to reach the pain threshold. In healthy control, that doesn't happen in chronic fatigue syndrome patients, where the threshold stays the same or even decrease a bit sometimes. And another important result, I think, is that we failed to set an experiment, the second one, a self-paced and physiologically limited experiment. We failed to set a test, an exercise that would not increase patient symptoms, because patients referred a worsening of their symptoms, and this is also reflected by PPTs after both type of exercises. Okay. Same thing with when we looked at the immune system changes. So immune system changes did not differ between groups, between patients and controls. Complement system significantly decreased after both type of exercises, and it did in both groups. No results in any way when we looked at elastase activity. So the only changes that we found was about complement system activation. That decreased in both groups in a similar way after both type of exercises. When we looked at association, so we performed some correlation analysis between complement system changes and PPTs, what we saw is that no association were found in healthy controls after the submaximal test or the self-paced exercise. And on the contrary, we did find positive correlation after both type of exercises in chronic fatigue syndrome patients. We also tried to perform regression analysis, and we found that complement system changes can explain a part, so around 24% of the change in PPTs. We tried also to discuss these results, and of course we are aware that this sample, we acknowledge that the sample was not large and that results were not straightforward, because we measured PPTs in 3 different parts of the body. We measured at the thumb and the back and at the calf. and we only found associations, moderate or strong association, only in 2 out of 6 measurements. And also we know that we had to use very indirect measures for both central sensitization or central nervous system excitability and for the immune system responses. But actually that limit could also be seen as strength of the results, because despite we used very indirect measures, we found some moderate or strong associations. So the more the immune system changes after exercise in chronic fatigue syndrome patients, the more the pain thresholds change as well. And I would like also to remark this, that is both type of exercise, despite the fact that the second one was meant to be of low demanding for patients, both type of exercise had similar effects and for patients. So that means that maybe it's not the type of exercise that matter for this kind of patient, but the exercise and physical activities itself can be a very stressful event for patients. Of course, we would like to thank all the people who participated, the researchers involved, EMA, EME Research UK to make this possible, and I thank you for your attention.
Great, thank you, and thank you for keeping to time. We now actually have a few minutes for questions, and we've got 2 microphones, and if you run out of questions for Andrea, then we'll allow you to ask questions for Andreas and Julius. Thank you. Any questions?
Can I just ask you, did you only look at C4A in terms of complement system?
Yep.
What are your plans to study complement system going forward then?
We are now actually looking at, you mean the next?
Yeah, it's just obviously C4A is only one part of the complement cascade, C3A, C5A as well, but also they're the activation products, the anaphylatoxins that produce inflammation. So it's only one very small part of the complement system. So do you have any plans to examine that further?
No, we don't have actually any plans to examine other things. When we had to choose, we searched the literature and we could find that the most studied proteins of the complement system were C3, C4, and C5 when we talk about pain, of course. And C3 and C5 were actually more associated than C4, but they were all animal studies. The only study that we found on humans found that actually C4 increased after exercise in chronic fatigue syndrome patients, but C3 and C5 didn't. So they explored the 3 proteins and they found only C4. That's why we decided to take the one as a measure.
Wait, there's a question over there. I saw several hands go up, but I can't remember where they were. Right, okay.
Hi there, it's Clive Card from me. Do you have any idea how the decreases in complement might actually be inducing the pain or changing the pain levels? You know, a mechanistic.
Well, so we tried to discuss that as well, because actually what we found is a decrease in complement system, and as I showed in the introduction, it generally increases in other studies. The way we try to explain this is that other studies looked at complement system changes after 6 hours after the exercise, and we measured it straight away after 1 hour after the exercise. So probably what is happening here is that the complement system has a quick decrease and then it rises up after a bit of time. And we think that can be in line with what we know about the immune system, that a lot of complex interaction happens and some part of the immune system decreases, some part of the adaptive immune system increase at the same time, and there are very complex interactions that we are not really able to explain measuring just one protein.